Hello, my name is Anurag. This is a poster I made as part of my part-time master's degree in neuroscience and neurodegeneration from the University of Sheffield. It explores the effects of the ApoE-ε4 genotype in Alzheimer's disease, with a focus on amyloid beta deposition. Just for some context, Alzheimer's disease is the most common form of dementia which can affect a person's cognition, memory, functional abilities, and behavior. Classically, there are two pathological hallmarks of this disease: amyloid beta plaques and neurofibrillary tau tangles. Amyloid beta, in particular, causes neurodegeneration by tau hyperphosphorylation, oxidative stress, and neuroinflammation. Apolipoprotein E is a compound which is primarily produced in the liver, but a more sialylated cerebral version is produced in astrocytes and microglia. Its main function is lipid transportation. The most common variant of ApoE is epsilon 3. The epsilon 2 allele has been shown to reduce Alzheimer's risk, while the epsilon 4 genotype can increase the risk of developing Alzheimer's by up to as much as a 15-fold effect when a person has two copies of this gene. In addition to this increased risk, ApoE4 carriers have higher amyloid beta loads and experience an accelerated burden of it. With ApoE4 being such a key risk factor for Alzheimer's disease, its array of pathological mechanisms have been the focus of numerous studies in the literature, as can be seen in Figure 1. This poster will focus primarily on amyloid beta-related pathways, which cause deposition of amyloid beta by reduced clearance and increased aggregation. As can be seen in Figure 2, apolipoprotein E is predominantly expressed in astrocytes, where ABCA1 lipidates it and shuttles it into the interstitial fluid. From here, it can be transported into cells via receptors like LRP1 and LDLR. Meanwhile, amyloid beta is produced in neurons. In ApoE's lipidated form, it can bind amyloid beta and facilitate its clearance. When ApoE is not functioning optimally, it can co-localize in plaques and affect amyloid fibrillation. In addition, suboptimal ApoE can affect amyloid clearance and aggregation, with some scant cell line-based evidence indicating it can affect amyloid production as well. ApoE4 is physically different from ApoE3 in that it contains an arginine amino acid at position 112 instead of cysteine due to a mutation on chromosome 19. This brings about numerous physical and functional changes. For example, a shape change in the ApoE4 exposes arginine at position 61 and leads to greater interaction between the N and C terminals of the protein. Functionally, ApoE4 is a poorer lipid carrier and forms larger co-aggregates when binding amyloid beta. It has more associations with VLDL and LDL receptors. It also leads to amyloid beta protein seeding, aggregation, and plaque formation and defective clearance. The mechanisms of the effect of ApoE4 on amyloid beta deposition can mostly be divided into reduced beta amyloid beta clearance and increased aggregation. Reduced clearance has been shown in a number of ways. One such study showed lower CSF amyloid beta concentrations in patients with two pairs of the ApoE4 alleles, indicating parenchymal accumulation due to impaired clearance into the CSF. Proposed mechanisms can be seen in Figure 3 and include inadequate enzymatic degradation of amyloid beta, poor receptor-mediated blood-brain barrier transportation, and impaired lipidation with dysfunctional endocytosis by glial cells. Increased amyloid beta aggregation in ApoE4 carriers was shown in a study on mice where amyloid beta deposition and neuritic dystrophy was enhanced during the seeding stage in amyloid transgenic mice when ApoE4 was overexpressed. The proposed mechanism here can be visualized using Figure 4. ApoE binding to an amyloid beta seed typically blocks fibrillogenesis. However, the clunky co-aggregates that ApoE4 tends to form behave differently in that they bind amyloid fibrils and promote plaque formation. In conclusion, ApoE4 has a significant impact on increasing amyloid beta deposition in Alzheimer's disease. These amyloid pathways are a key part of the nexus of changes that increase the risk profile for Alzheimer's disease, and future studies in this area may yield suitable therapeutics that target amyloid beta clearance or aggregation. My references can be accessed using the QR code below or the link below. Thanks very much.